Chapter 9 Review and Summary from OpenStax Book of Microbiology, which is free to everyone. Most bacterial cells divide by binary fission. Generation time in bacterial growth is defined as the doubling time of the population. Cells in a closed system follow a pattern of growth with 4 phases: lag, logarithmic, which is exponential growth, stationary, and death. Cells can be counted by direct viable cell count. The pour plate and spread plate methods are used to plate serial dilutions into or onto, respectively, agar to allow counting of viable cells that give rise to colony-forming units. Membrane filtration is used to count live cells in dilute solutions. The most probable cell number, MPN, method allows estimation of cell numbers in cultures without using solid media. Indirect methods can be used to estimate cell culture, the density by measuring turbidity of a culture, or live cell density by measuring metabolic activity. Other patterns of cell division include multiple nucleoid formation in cells, asymmetric division, as in budding, and the formation of hyphae and terminal spores. Biofilms are communities of microorganisms enmeshed in a matrix of extracellular polymeric substance. The formation of a biofilm occurs when planktonic cells attach to a substrate and become sessile. Cells in biofilms coordinate their activity by communicating through quorum sensing. Biofilms are commonly found on surfaces in nature and the human body, where they may be beneficial or cause severe infection. Here is a mozzie. My goodness. Aerobic and anaerobic environments can be found in diverse sites within and on the human body. Microorganisms vary in their requirements for molecular oxygen. Obligate aerobes depend on aerobic respiration and use oxygen as a terminal electron acceptor. They cannot grow without oxygen. Obligate anaerobes cannot grow in the presence of oxygen. They depend on fermentation and anaerobic respiration using a final electron acceptor other than O2. Facultative anaerobes show better growth in the presence of oxygen, but will also grow without it. Although aerotolerant anaerobes do not perform aerobic respiration, they can grow in the presence of oxygen. Most aerotolerant anaerobes test negative for catalase, which is an enzyme. Microaerophiles need oxygen to grow, albeit at a lower concentration than 21% of oxygen in air. Optimum oxygen concentration for an organism is the O2 level that promotes the fastest growth rate. The minimum permissive oxygen concentration and the maximum permissive oxygen concentration are respectively the lowest and highest O2 levels that an organism will tolerate. Peroxidase, superoxidase dismutase, and catalase are the main enzymes involved in the detoxification of the reactive oxygen species. Superoxide dismutase is usually present in the cell and can tolerate O2. All 3 enzymes are usually detectable in cells that perform aerobic respiration and produce more ROS. A capnophile is an organism that requires a higher than atmospheric concentration of CO2 to grow. Bacteria are generally neutrophiles. They can grow best at a neutral pH close to 7.0. Luna. Acidophiles grow optimally at a pH near 3.0. Alkaliphiles are organisms that grow optimally between a pH of 8 and 10.5. Extreme acidophiles and alkaliphiles grow slowly or not at all near a neutral pH. Microorganisms grow best at their optimum growth pH. Growth occurs slowly or not at all below the minimum growth pH or above the maximum growth pH. Microorganisms thrive at a wide range of temperatures. They have colonized different natural environments and have adapted to extreme temps. Both extreme hot and cold temps require evolutionary adjustments to macromolecules and biological processes. Psychrophiles grow best at the temperature range of 0 to 15, whereas psychotrophs thrive between 4 and 25. Mesophiles are in the middle range. They grow best at moderate temps in the range of 20 to 40. Pathogens are usually mesophiles. Thermophiles and hyperthermophiles are adapted to life at temps above 50. Adaptations to cold and hot temps require changes in the composition of membrane lipids and proteins. Halophiles require high salt concentration in the medium where the halotolerant organisms can grow and multiply in the presence of high salt but don't require it for growth. Halo is salt. Halotolerant pathogens are an important source of foodborne illnesses because they can contaminate foods preserved in salt. Photosynthetic bacteria depend on visible light for energy. Most bacteria, with a few exceptions, require high moisture to grow. Chemically defined media contain only chemically known components. Selective media favor the growth of some microorganisms while inhibiting others. Enriched media contains added essential nutrients that a specific organism needs to grow. Differential media helps distinguish bacteria by the color of the colonies or the change in the medium. Total plate count is used to measure the concentration of bacteria in something like processed peanut butter. The phase you would expect to observe the most endospores in a bacillus cell culture is the death phase. Penicillin, an antibiotic that inhibits cell wall synthesis, would be most effective in the log phase. Generation time is the length of time it takes for a population of cells to double. The function of the Z ring in binary fission is to form a contractile ring at the septum. If a culture starts with 50 cells, it will have, doubling each time, 1,600 cells after 5 generations without cell death. For example, filamentous cyanobacteria often divide by fragmentation. Antimicrobial resistance is higher in a biofilm than a free-floating bacterial cells due to cells being metabolically inactive at the base of the biofilm. Quorum sensing is used by bacterial cells to determine the density of the population. Autoinducers bind directly to DNA to activate transcription. An inoculated thioglucolate medium culture tube shows dense growth at the surface and turbidity throughout the rest of the tube. It is likely that the organisms are facultative anaerobes. An inoculated thioglycolate medium culture tube is clear throughout except for dense growth at the bottom of the tube. It is likely that the organisms are obligate anaerobes. Pseudomonas aeruginosa is a common pathogen that infects airways of patients with cystic fibrosis. It doesn't grow without oxygen. It is probably an obligate aerobe. Aerobe needs oxygen. Streptococcus mutans is a major cause of cavities and resides in gum pockets. It doesn't have a catalase activity and can be grown outside of an anaerobic chamber. It's likely an aerotolerant anaerobe. Neisseria gonorrhea is recommended to grow in a CO2-enriched environment as it's a capnophile. Bacteria that grows well in mine drainage, where the pH is 1 to 2, are likely acidophiles. A particular lake where the pH is close to 10 grows a bacteria likely to be an alkaliophile. An acidophile would likely thrive in a pH 1.5. Contaminated contents in a refrigerated environment would likely be contaminated with psychrotrophs, which like cold places. A hot tub about 37 degrees, the bacteria could be mesophiles. A hyperthermophile would thrive in an environment like a hydrothermal vent at the bottom of the ocean. a mountain lake of 12 degrees would likely harbor psychrophiles. The reason that jam and dried meats don't require refrigeration to prevent spoilage is because they have a low water activity. Bacteria living in salt marshes are most likely to be halophiles. Chapter 13, summary with question and answer review from OpenStax Microbiology book, which is free to everyone. Inanimate objects that may harbor microbes and aid in their transmission are called fomites. The level of cleanliness required for a fomite depends on both the item's use and the infectious agent with which the fomite may be contaminated. The CDC and the NIH have established 4 biological safety levels, BSLs, for labs performing research on infectious agent. Each level is designed to protect lab personnel and the community. The BSLs are determined by the agent's infectivity, ease of transmission, and potential disease severity, as well as the type of work being performed by the agent. Disinfection removes potential pathogens from a fomite, whereas antisepsis uses antimicrobial chemicals safe enough for tissues like skin. In both cases, microbial load is reduced, but microbes may remain unless the chemical is strong enough to be a sterilant. The amount of cleanliness, sterilization versus high-level disinfectant versus general cleanliness, required for items used clinically depends on whether the item would come in contact with sterile tissues, critical items, or mucous membranes, semi-critical items, or intact skin, non-critical items. Medical procedures with a risk for contamination should be carried out in a sterile field maintained by proper aseptic technique to prevent sepsis. Sterilization is necessary for some medical applications, as well as in the food industry, where endospores of Clostridium botulinum are killed through commercial sterilization protocols. Physical or chemical methods to control microbial growth that result in the death of the microbe are indicated by the suffixes "side" or "sidal", whereas those that inhibit microbial growth are indicated by suffixes of "stat" or "static", like stasis. Microbial death curves display the logarithmic decline of living microbes exposed to a method of microbial control. The time it takes for a protocol to yield 1 log, 90% reduction, in the microbial population is the decimal reduction time, or the d-value. When choosing a microbial control protocol, factors to consider include the length of exposure time, the type of microbe targeted, its susceptibility to the protocol, the intensity of the treatment, the presence of the organics that may interfere with the protocol, and the environmental conditions that may alter the effectiveness of the protocol. Heat is a widely used and highly effective method for controlling microbial growth. Dry heat sterilization protocols are commonly used in aseptic techniques in the lab. However, a moist heat sterilization is typically the more effective protocol because it penetrates cells better than dry heat does. Moist heat is better than dry heat. Pasteurization is used to kill pathogens and reduce the number of microbes that cause food spoilage. High-temp, short-time pasteurization is commonly used to pasteurize milk that will be refrigerated. Ultra-high temp can be used for milk that will be in long-term storage unrefrigerated. Refrigeration slows microbial growth. Freezing stops growth, killing some organisms. Lab and medical specimens may be frozen on dry ice or at ultra-low temps for storage and transport. High-pressure processing can be used to kill microbes in food. Hyperbaric oxygen treatment to increase oxygen saturation has also been used to treat certain infections. Desiccation has long been used to preserve foods and is accelerated through the addition of salt or sugar, which can both decrease water activity in foods. Lyophilization combines cold exposure and desiccation, freeze-drying, for long-term storage of foods and lab materials, but microbes remain and can be rehydrated. Ionizing radiation, including gamma radiation, is an effective way to sterilize heat-sensitive and packaged materials. Non-ionizing radiation, like UV light, is unable to penetrate surfaces but is useful for surface sterilization. HEPA filtration is commonly used in hospital vent system and biological safety cabinets in labs to prevent transmission of airborne microbes. Membrane filtration is commonly used to remove bacteria from heat-sensitive solutions. Heavy metals, like mercury, silver, and copper and zinc, have long been used for disinfection and preservation, although some have toxicity and environmental risks associated with them. Halogens, including chlorine and fluorine and iodine, are also commonly used for disinfection. Chlorine compounds, including sodium hypochlorite, chloramines, and chlorine dioxide, are commonly used for water disinfection. Iodine, in both tincture and iodophore forms, is an effective antiseptic. Alcohols, including ethyl alcohol and isopropyl alcohol, are commonly used as antiseptics that act by denaturing proteins and disrupting membranes. Phenolics are stable, long-acting disinfectants that denature proteins, disrupt membranes. They are commonly found in household cleaners, mouthwash, and hospital disinfectant, and are also used to preserve harvested crops. The phenolic compound, triclosan, found in antibacterial soaps, plastics, and textiles, is technically an antibiotic because of its specific mode of action of inhibiting bacterial fatty acid synthesis. Surfactants, including soaps, detergents, lower the surface tension of water to create emulsions that mechanically carry away microbes. Soaps are long-chain fatty acids, whereas detergents are synthetic surfactants. Quaternary ammonium compounds, or QUATs, are cationic detergents that disrupt membranes. They are used in household cleaners, skin disinfectants, oral rinses, and mouthwashes. Bisbiguanides disrupt cell membranes, causing cell contents to gel. Chlorhexidine and alexidine are commonly used for surgical scrubs, hand washing in clinical settings, and in prescribed oral rinses. Alkylating agents effectively sterilize materials at low temps, but they are carcinogenic and may also irritate tissue. Glutaraldehyde and ofaldehyde are used in hospital disinfectants, but not as antiseptics on people. Formaldehyde is used for storage of tissue specimens as an embalming fluid and in vaccine preparation to inactivate infectious agents. Ethylene oxide is a gas sterilant that can permeate heat-sensitive packaged materials, but it is also explosive and carcinogenic. Peroxygens, including hydrogen peroxide, peracetic acid, benzoyl peroxide, and ozone gas, are strong oxidizing agents that produce free radicals in cells, damaging their macromolecules. They are environmentally safe and are highly effective disinfectants and antiseptics. Pressurized carbon dioxide in the form of a supercritical fluid easily permeates packaged materials and cells, forming carbonic acid and lowering intracellular pH. Supercritical carbon dioxide is non-reactive, non-toxic, non-flammable, and effective at low temps for sterilization of medical devices, implants, and transplanted tissue. Chemical preservatives are added to a variety of foods. Sorbic acid, benzoic acid, propionic acid, and their more soluble salts inhibit enzymes or reduce intracellular pH. Sulfites are used in winemaking and food processing to prevent browning of foods. Nitrites are used to preserve meats, maintain color, but cooking nitrite-preserved meats may produce carcinogenic nitrosamines. Lovely. Niacin and natamycin are naturally produced preservatives used in cheese and meats. Nisin is effective against gram-positive bacteria and natamycin against fungi. Chemical disinfectants are grouped by the types of microbes and infectious agents they are effective against. High-level germicides kill the vegetative cells, fungi, viruses, and endospores, and can ultimately lead to sterilization. Intermediate-level germicide cannot kill all viruses and are less effective against endospores. Low-level germicides kill vegetative cells and some enveloped viruses, but they are ineffective against endospores. The effectiveness of a disinfectant is influenced by the length of exposure, concentration of disinfectant, temp, and pH. Historically, the effectiveness of a chemical disinfectant was compared with that of a phenol at killing Staphylococcus aureus and Salmonella enterica, Serova typhi, and a phenol coefficient was calculated. The disc diffusion method is used to test against the effectiveness of a chemical disinfectant against a particular microbe. The use dilution test determines the effectiveness of a chemical disinfectant on a surface. Chapter 16, Summary, from OpenStax Microbiology, which is free to everyone. Epidemiology is the science underlying public health. Morbidity means being in a state of illness, whereas mortality refers to death. Both morbidity and mortality rates are of interest to epidemiologists. Incidence is the number of new cases, morbidity or mortality, usually expressed as a proportion during a specified time period. Prevalence is the total number affected in the population, also usually expressed as a proportion. Sporadic diseases only occur rarely and largely without a geographical focus. Endemic diseases occur at a constant and often low level with a population. Epidemic diseases and pandemic occur when an outbreak happens on a significantly larger than expected level, either locally or globally. Koch's postulates specify the procedure for confirming a particular pathogen as the etiologic agent of the disease. Koch's postulates have limitations of the application if the microbe cannot be isolated and cultured, or if there is no animal host for the microbe. In this case, molecular Koch's postulates would be used. In the USA, the CDC monitors notifiable diseases and publishes weekly updates on the Morbidity and Mortality Weekly Report. Early pioneers of epidemiology such as John Snow, Florence Nightingale, and Joseph Lister studied disease at the population level and used data to disrupt disease transmission. Descriptive epidemiology studies rely on case analysis and patient history to gain information about outbreaks, frequently while they are still occurring. Retrospective epidemiology studies use historical data to identify associations with the disease state of present cases. Prospective epidemiology studies gather data and follow cases to find associations with future disease states. Analytical epidemiology studies are observational studies that are carefully designed to compare groups and uncover associations between environmental or genetic factors in disease. Experimental epidemiology studies generate strong evidence of causation in disease or treatment by manipulating subjects and comparing them with control subjects. Reservoirs of human disease can include the human and animal populations in soil, water, and inanimate objects. Contact transmission can be direct or indirect through a physical contact with either an infected host, which is direct, or contact with a fomite that an infected host has made contact with previously, indirect. Vector transmission occurs when a living organism carries an infectious agent on its body, which is mechanical, or as an infectious host itself, which is biological, to a new host. Vehicle transmission occurs when a substance such as soil, water, or air carries an infectious agent to a new host. Healthcare-associated illnesses, or also called nosocomial infections, are acquired in the clinical setting. Transmission is facilitated by medical interventions and the high concentration of susceptible immunocompromised individuals in clinical settings. The World Health Organization, the WHO, is an agency of the United Nations that collects and analyzes data on disease occurrence from member nations. The WHO also coordinates public health programs and responses to international health emergencies. Luna, stop that. Emerging diseases are those that are of new to human populations or that have been increasing in the past 2 decades. Re-emerging diseases are those that are making a resurgence in susceptible populations after previously having been controlled in some geographical areas. This was read by me, for me, so I can understand microbiology. Thank you.